All right. The purpose of this video is to give my biology students an opportunity to practice with questions that are going to resemble questions like they had. Hold on. I can turn off my video now. Don't need the video. All right. To give my students an opportunity to practice with the types of questions they will see on the final. Best way to practice with this video and with these questions is with a study guide, as information comes from the entire semester and there are a lot of notes you have to pull from. So hopefully you got your study guide organized and ready to go. Also, just for giggles, if you would like, keep track of how many of the questions you get correct. We've got 6, 18, 36, 44, 48. We have 51 total questions. See how many out of the 51 questions you can get correct. Give me 1 second. I'm going to close the door. My teenager is thumping his music next to me. All right, here we go. I am going to speed through this a little bit. For the 1-point questions, I'm going to be doing a timer of about a minute. The 2-point questions, I'll be doing a timer of about 1 minute and 45 seconds. 3 and 4, we'll do just slightly longer. Let me adjust this to 2:30. Okay, bam. All right, here we go, kids. What is the correct order of the scientific method? Hint, there are 5 steps from start to finish. Okay, scientific method, not to be confused with the steps in a lab report. The scientific method, these would be in your scientific method class notes that we did way back at the beginning of the semester. Also, don't forget Biology World. If you have any questions for me or you need to speak about anything between now and Friday, just send me an email and I will make some time for you. All right, here we go. You got to identify the problem, form a hypothesis, perform the experiment, analyze your data, and then you have 2 choices. You either modify the experiment if your data does not match what your hypothesis was, or if it does, then you communicate results. All right, here we go. Here is the grid. Okay, this is the base units, or pardon me, these are the base units, and these are the prefixes. Convert this to meters and convert this to normal notation. Give you guys a couple minutes to work on that one. So remember, you move the decimal the same direction as the prefix. So we got kilo, which is way over here. Meters is right here in the middle. So you're moving the decimal to the right, and you just got to figure out how many spaces to the right you need to move it. Second one, this is very, very similar. Yesterday's warm-up question. What does a positive exponent mean? And what does a positive 3 exponent mean? All right, about 30 seconds. All right, here we go. Okay, so we start at kilo, we end up at meter, we go 1, 2, 3 spaces to the right. Therefore, 1, 2, if you run out of numbers, you add a 0, so that's going to be 7320 meters. Okay, positive exponent means we move to the right, and then the exponent itself tells you how many spaces to the right. 1, 2, 3, 7746.3. You'll need to know how to do both of those types of things on your final. The process used to find answers to questions. What do we call that process? What's the correct order of the sections in a lab report? Wasn't that yesterday's warm-up question? Data with units, numbers, and that is measured is what kind of data? There are two types of data. There's qualitative data and there's quantitative data. You'll need to know which one has units, numbers, and is measured. All right, about 45 more seconds. And again, I know that many of you have these answers already, and you can do it in quicker than a minute. But for those people that are actually using this as an opportunity to do some studying, finish up their study guide, they might need a little bit more time. All right, you guys, here come the answers. Okay, the scientific method is a process used to find answers to questions. Title, objective, hypothesis, procedure, data, and conclusion. And data that has numbers, units, and needs to be measured is quantitative. Easiest way to remember is quant count. Quant count. All right, here we go. What are the 3 goals of science? The variable that is deliberately changed in an experiment is which variable? All right, about 40 more seconds. 3 Goals of Science, that's in section 1-1 of your book. And I'm pretty sure 1-2 has the experiment information. All right, here we go. Okay, goals of science: investigate and understand the natural world, explain events in the natural world, or use derived explanations to make useful predictions. And there's 2 ways you can describe this variable: the manipulated or independent variable. And the easiest way to remember this is the independent variable is the one that I change. Okay. Here we go. On to organic molecules, Unit 2. What are the 3 particles that make up an atom? All right, here we go. Protons are positive, electrons are negative, neutrons are neutral, like Switzerland. All right, here we go. What are all of the elements of life found in carbohydrates, lipids, proteins, and nucleic acids? So every element that is involved in those compounds and molecules, what are they? Which organic molecule is the main source of immediate energy? All right, about 30 seconds. All right, here we go. Hydrogen, carbon, oxygen, all found in carbohydrates. Nitrogen, found in nucleic acids, also found in proteins. Phosphorus, found in nucleic acids. Sulfur, found in proteins. Notice there are some elements missing, not missing from this list. Therefore, if you see an element that is not part of this list on the test, that is the one that is not an element of living things. And quick energy source, we're talking carbohydrates, we're talking breads, tortillas, candy, sugar, all that stuff. All right, here we go. Which organic molecule stores genetic information as a genetic code? What are the monomers of proteins? And what type of organic molecule is this? Remember, there's only 4 types of organic molecules: carbohydrates, lipids, proteins, and nucleic acids. Give you guys a couple minutes to look over this. All right, here we go. Okay, nucleic acids, DNA stores genetic information as a genetic code. Also, a question that many of you guys missed on the DNA replication lab, it was that hard question, number 17, that I couldn't give you too many clues. The actual code is the order of nitrogen bases. So, for example, and you'll learn this next semester, that sequence AGGTCAGTTGAAACTGG, whatever that code is, that will code for specific proteins. So it's the order of the nitrogen bases that determine what proteins you have. And one human to another, our DNA is 99.99% identical, but that 0.01% in 80,000 genes accounts for a wide variety of variation. It's why I have green eyes and you have blue eyes. It's why, I'm 5'7" and stocky and you're 6'1" and slim. It's why I have brown hair and you have blonde hair. It is all tied to the order of nitrogen bases on our DNA molecules. Monomers of proteins are amino acids, and this is a lipid. This is glycerol in blue, and these are the fatty acids here in black. Come on. I dislike this thing. I'm going to put you up there. Okay, here we go. All right, onward. Glucose and fructose are both examples of what type of organic molecule? C6H12O6 and H2O are both blanks. Sketch the structure of a carbohydrate. You probably won't have an opportunity to do that, but locate it on your study guide, hopefully. And blanks are made of monomers called amino acids. Are you kidding me? We just talked about that. Give you guys a couple minutes to think about that. Oh, whoa. Hold on. Back. My bad. That's what I get for multitasking. Ay. That was stressful. All right, a couple more seconds. All right, ladies and gentlemen, here we go. Glucose and fructose, anything that ends in OSE, lactose, maltose, dextrose, those are all carbohydrates. All right, C6H12O6 and H2O are both molecules or compounds. Technically, a molecule only has one atom or one element, just more than one atom of that element. But we kind of use compounds and molecules interchangeably. Technically, a compound is two different elements, which are both of those, but sometimes we call them molecules. Anyway, let's see. This is a carbohydrate. Remember, you're going to have a carbon backbone. Sometimes this will be in a ringed structure, and you're going to have hydrogen and hydroxides off of the center carbon chain, and you will always have at least one double bond to an oxygen. And then let's see, proteins are made of monomers called amino acids. Second time we've seen that. That may or may not mean that there's more than one question that requires you to know amino acids make proteins. All right, here we go. On to ecology and now let's do some random questions. Let's jump around a little bit. Phosphate groups, guanine and cytosine, are both found in what and what? About 30 seconds. All right, here we go. Notice there's no thymine in there. There's no deoxyribose in there. And there's no ribose in there because those are found in only one type of molecule. These three are found in both types of molecules. Which scientist was recognized for taking an X-ray diffraction image of DNA? Famous photo 51. All right, a couple more seconds. Yep. All right, possibly, I don't know, maybe one of the most famous female scientists of all time for her work that led to understanding the structure of DNA, and totally got the shaft from Wilkins, Watson, and Crick, who barely mentioned her when they got their Nobel Prize. All right, here we go. What are 3 similarities and 3 differences between DNA and RNA? And let me take care of this typo there. Three similarities, 3 differences between DNA and RNA. These are in the 12-2 class notes. About 30 more seconds. All right, ladies and gentlemen, here we go. Okay, similarities. They're both nucleic acids, RNA, DNA, Na. They're both made from nucleotide monomers. They both have cytosine, adenine, guanine, and they both have a sugar phosphate backbone. Differences: DNA is double stranded, RNA is single stranded. DNA has the sugar deoxyribose, RNA has the sugar ribose. DNA has the nitrogen based thymine, RNA has the nitrogen based uracil. Another difference: DNA has to stay in the nucleus, RNA can be in the nucleus and in the cytoplasm. I think there's like one more difference, but if you know those 3 or 4, you will be fine. Here we go. A little bit more random. A solution with a pH above 7, what do we call that? Solution with a pH below 7, what do we call that? A solution with a pH equal to 7, what do we call that? About 30 seconds. All right, here we go. Okay, pH above 7, and this is where a lot of kids get confused. They think that a higher number means it's acidic. No, no, no. 7.0001 all the way up to 14 is base, basic alkaline. Acidic numbers are basically 0.00001 all the way to 6.9999999. Those are acidic. And then, of course, neutral is equal amounts of hydrogen and hydroxide ions. Let's talk about that for a second. You don't need to know this for the final, but it's a good review anyway. Remember, hydrogen atoms, H plus atoms, hydrogen ions, okay? The more hydrogen ions you have, the more acidic it is. So if you have a pH of 2, you're going to have more hydrogen ions than hydroxide. Whereas if we're talking about a base and we're talking about a pH of 10, you will have more hydroxide ions than you have hydrogen ions. And of course, if we're talking about a pH of 7, neutral, we're going to have equal amounts of hydrogen and hydroxide ions. What are organisms that eat both plants and animals? Like most humans eat both plants and animals. The repeated movement of water between Earth's surface and the atmosphere, what do we call that? All right, about 30 seconds. All right, here we go. Okay, omni is the Latin root for all. Okay, so omnivores eat plants and animals, herbivores eat plants only, carnivores eat animals only. Okay, omnivores eat both, and most humans, like I said before, are omnivores. And then the water cycle, how water moves through the different spheres: the hydrosphere, the atmosphere, the biosphere, the lithosphere. What sphere am I missing? I forget what the other sphere is. Okay, you guys get the point. Okay, here we go. Another random question. If an enzyme's activity slows down as temperature increases, what is likely happening to the enzyme? What do we call that when an enzyme gets damaged because of heat? The original source of almost all energy in most ecosystems, what do we call that source? A couple more seconds. All right, here you go. So we call that denaturization, but it basically means it's changing the three-dimensional structure. Remember, enzymes have a lock and key fit with the substrate. So if you change the lock, the key is no longer going to work. So they basically are getting damaged or destroyed. And sunlight is the original source of almost all the energy in most ecosystems. There is some chemical energy that happens in certain environments, but you can assume that everything else starts with photosynthesis and the sun. The diagram below shows the relative amount of blank available in an ecosystem. Sugar and phosphate form the blank of DNA. What types of bonds connect the nucleotides or nitrogen bases in the center of the DNA double helix? So, clue for the first question. So, what are these percentages referring to? You'll notice 10% is only 10% of 100. 1% is 10% of 10. 0.1% is 10% of 1. And of course, if we went another step up to another apex predator, this would be 0.01%. All right, here we go. Okay, so it's the energy. Okay, so we have at the producer level, we have 100% of the energy available, but herbivores or omnivores, they only get 10% of that available energy because 90% gets lost as heat. Same thing. And that's why top or apex predators have to eat a lot because what they are getting has lost a lot of energy through feeding relationships. Okay, sugar, phosphate backbone of the DNA molecule, and weak hydrogen bonds link the nitrogen bases in the DNA molecule. Okay, let's talk about climate and ecology. Sketch a food chain with one producer and 3 consumers. Remember, a food chain is just a line. Okay. A bunch of different food chains together in an ecosystem is a food web. Just do a food chain. And of course, on the test, you'll have to recognize which one is the producer, which ones are the consumers. All right, here we go. All right, so you're always going to start with some sort of a producer, in this case, grass. All right, mice eat grass. Mice also eat insects, so technically they would be an omnivore. Snakes eat mice. Snakes pretty much are going to be carnivores. I mean, it's possible a snake might munch on some grass every once in a while, but they're really good at catching insects and other things. So they most likely going to be carnivore. And then, of course, eagles and most raptors are going to be top level consumers. They are a carnivore as well. All right, here we go. An herbivore is an organism that eats what? What natural process releases carbon dioxide into the atmosphere? All right, about 30 seconds. And remember, if these questions are easy for you, the test should be easy for you as well. You will see what the correct answer is in the form of a multiple choice question. You do not see what the answer is. It's either you know it or you don't in Jeopardy. So if you're feeling pretty confident, the test should go well for you. All right, here we go. Answers are forthcoming. Herbivore. What? Oh, this is wrong. My bad. Hold on a second. An herbivore is an organism that eats plants, not. Oh, my bad. Yeah, they eat producers. Okay, we're just using the scientific word for it. What is going on? Hold on a minute. Okay. Let's go back in here. Yeah, so they eat producers, plants, or autotrophs. And respiration, us exhaling, is adding carbon dioxide to the atmosphere. And remember, respiration and carbon dioxide and water vapor come from a process called cell respiration. Cells in the presence of oxygen, that's why we need to breathe in oxygen, and food, primarily glucose, and that's why we need to eat, can get what is called ATP, energy for cells. Okay, and the product of that reaction is carbon dioxide and water vapor, which we exhale, and that's coming from our cells that need to get the energy. Blanks help in the nitrogen cycle by releasing nitrogen in animal waste. What do we call those organisms that release nitrogen in animal waste? What process speeds up the release of carbon from fossil fuels into the atmosphere? True or false? Matter decreases over time? Okay, about 30 seconds. All right, here we go. Okay, so decomposers break down organic molecules and they return nitrogen into the atmosphere, or pardon me, into the nature. Okay, sometimes it's atmosphere, sometimes it's actually the ground. Combustion, which is a fancy word for burning, okay, and matter does not decrease over time. It's called the law of conservation of matter. Matter can only be transformed or changed, but you can't like get rid of an atom. It just recombines with something else. So that is a false statement. All right, we got to speed this up. We got about 9 minutes until we got to end this meeting because I got another meeting starting right at 2. The lengths of days differs throughout the year because the blank of Earth is tilted. What? Right. What branch of biology deals with the interactions between organisms and their environment? What type of organism supply food to ecosystems? That would be what types of organisms supply food to ecosystems. Only about 10% of the available energy in a lower trophic level gets transferred to the next level. What happens to the other 90%? Give you guys a couple minutes to think about this one. Okay, about 15 more seconds. All right, here we go. It's the axis of Earth is tilted 23.5 degrees. That is the study of ecology. Producers, autotrophs provide the energy for food, and that 90% gets lost as heat, specifically metabolic heat. Okay, here we go. We need to get through these questions so we can get you guys out of here. DNA replication. What organic molecule is pictured below? And what do we call this shape? About 30 seconds. All right, 5, 4, 3, 2, 1. I know, quickest 30 seconds ever. All right, here we go. That is what? DNA, and we call that the double, the double helix. All right. Avery's experiments showed that bacteria are transformed by blank. Remember, Griffith's experiments led Avery to doing some different things with this molecule to figure out which one does the transforming in eukaryotes. Where do we find the DNA? Where do we find the chromosomes in eukaryotes? We are eukaryotes. Complex organisms are eukaryotes. All right, here we go. All right, so Avery proved DNA was the transforming factor built on the information that Griffith exposed to the world. And DNA is found in the nucleus. Chromosomes are found in the nucleus in eukaryotic organisms. Last questions, kids. What? This link is wrong. Don't worry about that first question. If DNA strand 1 is AACGCT and is being used in replication, what is the complementary strand? Use your base pairing rules. And after blank blank, each new molecule of DNA will have one old strand and one new or complementary strand. What do we call that process? Again, please ignore this top question. Matter of fact, I'm just going to do that now. This is going to be something we are learning next semester. Okay. So sorry about that. Okay, about 45 seconds. And actually, I will make this, sorry, I'm kind of a perfectionist when it comes to this type of thing. It's going to make it a 2-point question. Okay, here we go. Using base pairing rules, A, T, C, G, G, C, C, G, and T, A. Sorry, let me fix this. Get rid of that question. Again, I know you guys probably don't care about this. I do care about this. All right, and let's get rid of this question. Don't want to have any confusion when kids are watching this video later. Bam, 2, and one more thing to change. All right, ladies and gentlemen, that represents about 98% of the questions that you will see on this test. If you know the answers to many or most of these questions, you are absolutely going to hammer this test. Thanks for coming to the Zoom review. Don't forget, if you have any questions over the coming days, just send me an email and I'll set up some time to meet with you privately. All right, you guys, take it easy and we will see you soon.